Welcome back to the deep end. Today we're heading out into the open air. We're walking through the pasture, pushing through the tall grass, and we're looking for something that is, well, it's arguably the most iconic living thing in the entire fungal kingdom.
It really is.
I mean, you know exactly what I'm talking about. If you gave a 5-year-old a crayon and a piece of paper and said, "Draw a mushroom," this is what they would draw.
It's the archetype. 100%.
Exactly. It's the platonic ideal of a mushroom. You've got the white cap, a little skirt on the stem, and those pink gills underneath. It's the meadow mushroom, Agaricus campestris.
Or at least that's we've been calling it for nearly 300 years.
Right, and that is where this deep dive gets incredibly messy and, for me, incredibly fascinating. Because this mushroom, this one that looks like the wild, rugged cousin of the button mushroom you buy on a pizza.
Yeah, the one from the grocery store.
It's effectively the everyman of the mushroom world. It grows on lawns, it pops up in parks, it seems so friendly and so approachable. But as we dug into the research for this, from the taxonomy reports to the genetic sequencing data, we realized this simple mushroom is actually a total chaotic mess of hidden identities.
It is a taxonomic nightmare. And it's also, I think, a masterclass in how much we really don't know about the biology right under our feet.
We're going to find out that if you are hiking in North America and you spot a meadow mushroom, you are almost certainly not looking at Agaricus campestris.
Not even close, probably.
You're looking at an imposter. We're going to talk about brand new species that were discovered in China as recently as 2024, and that's just months ago, that are completely rewriting the family tree.
And we have to talk about the stakes.
Yeah.
Because, usually when we nerd out about taxonomy on this show, the worst thing that happens if you get it wrong is you look a little silly in front of a scientist.
Right.
Here, if you get the identification wrong, you might be inviting a very, very nasty character to dinner.
The evil twin.
The yellow stainer. Agaricus xanthodermus.
Which sounds like a spell from a fantasy novel, but it is actually a recipe for a weekend spent in the bathroom praying for the sweet release of, well, let's just say it causes severe gastrointestinal distress.
And that is putting it very, very mildly.
So our mission today is to decode the Agaricus genus. We are going to break down the biology of the fairy ring. We're going to navigate this complex new taxonomy that is literally being written as we speak, and master the critical safety checks, the smell and the stain that separate a delicious meal from a hospital visit.
And I think we have to start with the name itself, the history, because this mushroom, it has a pedigree.
It really does. We have to go back to the guy who started it all.
Linnaeus.
Carl Linnaeus, the father of modern taxonomy. So in 1753, he is going through nature and giving everything a two-part Latin name, this binomial nomenclature. And he gets to this common white field mushroom and calls it Agaricus campestris.
Which is a very literal name. I mean, campestris just translates to of the fields.
Which makes perfect sense. You don't find these guys deep in the dark, scary woods, huddled under a pine tree. You find them out in the sunlight. They are grassland specialists.
Exactly.
Yeah.
And for centuries, that was the assumption. If you were in Europe, Asia, North Africa, Australia, New Zealand, or North America, and you saw a white mushroom with pink gills growing in a field, it was Agaricus campestris. Case closed. It was considered a globally distributed species.
It was the Coca-Cola of mushrooms,
Right.
available everywhere.
Pretty much. And physically it is, or was thought to be, very distinct. If you are standing over one looking down, you are seeing a cap that is silky and white. It can be anywhere from, say, 3 to 12 centimeters across, so about an inch to maybe 4 or 5 inches.
And the shape changes, right?
It does. When it's a baby, what we call a button, it's hemispherical, almost like a little snowball. Then as it ages and opens up to drop its spores, it flattens out. Sometimes it gets so flat it's like a dinner plate.
But the real tell, the thing that makes a forager's heart skip a beat, is what happens when you flip it over.
The gills.
The gills.
The gills are the key. And in the entire genus Agaricus, the gills are what we call free.
What does that mean exactly? Free.
It means if you look really closely at where the gills meet the stem, they don't actually touch it. There's a tiny gap, like a little moat, around the top of the stem. They're unattached.
And the color. This is the most beautiful part, I think.
It is stunning. In a young, fresh specimen, those gills are a vivid, beautiful pink. The sources describe it as salmon or mauve. It's not a hot neon pink, but this really fleshy, earthy pink.
Yeah.
And this is crucial because it tells you the spores haven't matured yet. As the mushroom ages and the spores ripen, that pink darkens. It goes to a sort of red-brown, and finally, in an old specimen, it's a dark, rich chocolate brown.
I want to pause on that for a second because that color progression, pink to brown, is such a critical safety feature. We'll get into the deadly lookalikes later, but a lot of the things that will absolutely kill you, like the Amanitas,
Right.
they have white gills.
Yeah.
And they stay white forever.
That is the first rule of survival in the field. If you have what looks like an Agaricus, but it has white gills, you walk away fast. You absolutely need that pink to brown progression.
And then there's the stem, the stipe.
The stipe, yep. It's white, it's pretty sturdy, and it has that little ring on it, the annulus.
Which is basically a little skirt, right?
Exactly. That ring is just a remnant of what we call a partial veil.
Mm.
When the mushroom was a baby button, there was a thin membrane covering those delicate pink gills, protecting them. As the cap expanded and opened up like an umbrella, that membrane tore, and the ring is just what's left of it clinging to the stem.
Ecologically, these guys are like the good neighbors of the pasture. They aren't parasites. They're not killing trees.
No, not at all. They're sampratroughs. They're the cleanup crew. Their job is to feed on dead organic matter. So they're decomposing the thatch and the grass. They're breaking down dead roots. And let's be honest, they're breaking down manure.
They do love a horse pasture.
They love nitrogen. And horses are very, very good at providing, let's call it nitrogen-rich fertilizer.
So, looking at the IUCN Red List data we have, these mushrooms are officially considered least concern. The populations are large and stable globally. But I read something interesting about local declines.
Yes, in areas where traditional agriculture is changing, specifically where horse pastures are being converted into, say, cornfields or housing developments, the local populations of Agaricus plummet. They really need that specific grazing ecosystem to thrive.
Okay, so that's the classic picture, the friendly field mushroom. But before we get into the massive identity crisis this mushroom is suffering from, I want to talk about how they grow. Because you mentioned they grow in fields, but they don't just pop up randomly like confetti. They have a very specific, very beautiful geometry.
The fairy ring.
The fairy ring. It sounds magical. It sounds like something out of folklore where you step inside and, lose 20 years of your life or get whisked away by elves. But it's biological. Why a circle? Why not a square or a random patch?
To understand the ring, you have to completely shift your perspective. The mushroom you see above ground, the white cap, the stem, that is just the fruit. That's the apple on the tree. The tree itself, the main body of the organism, is underground, is the mycelium.
The network of threads.
Right. So imagine a single microscopic spore lands in a perfect spot in a grassy field. It germinates. It sends out these tiny microscopic threads called hyphae. This forms what we call the primary mycelium. Now, here's where it gets a little bit romantic, actually.
Okay.
This primary mycelium is haploid. It only has half the genetic material it needs to reproduce.
It's incomplete. It's looking for a partner.
Exactly. It's like someone on a dating app. It's short-lived, it's pretty fragile, it's frantically searching through the soil for another hypha from a different but compatible spore.
Yeah.
When they finally meet, they fuse. This process is called plasmogamy.
And then they settle down.
Then they form the secondary mycelium. And this is the real powerhouse. It's dekaryotic, which is a fancy way of saying it has two different nuclei in each cell, one from each parent. This is the married couple. It's robust, it's long-lived, and it's ready to build an empire underground.
And that empire grows outward.
Radially. Think of a ripple in a pond when you throw a stone in. The soil in a lawn is usually pretty uniform, right? So as the mycelium consumes all the nutrients at the center where it started, it depletes them. It has to keep moving outward to find more food, so it grows year after year in a perfect circle.
So the dead spot in the middle of a fairy ring is where the fungus started maybe decades ago?
Decades, or even centuries. There are fairy rings in England that are estimated to be over 700 years old. They're ancient organisms. The mycelium is expanding at what we call the leading edge. That's where the energy is. That's where the active digestion is happening. So when the conditions are just right, usually a drop in temperature and a good soaking rain, the fungus decides it's time to reproduce. And it sends up its fruit bodies, the mushrooms, right at that active edge of the colony.
Which creates a perfect ring of mushrooms. But I've noticed something else about these rings. Even when the mushrooms aren't there, you can still see the circle in the grass.
You see the color change, right?
Yeah, there's this band of grass that is taller, darker green, and just looks way more lush than the rest of the lawn.
That is the wood wide web acting as a fertilizer factory.
Yeah.
As the mycelium breaks down all that organic matter at that leading edge, it's releasing a massive amount of nitrates and ammonia right into the soil. It's effectively dumping Miracle-Gro on the grass roots. So the grass responds by, hulking out.
But I've also seen the opposite, a dead zone. A ring of brown, totally dead grass.
Yeah, that happens when the mycelium gets too successful for its own good. The fungal mat becomes so dense and thick underground that it actually becomes hydrophobic. It starts to repel water. It creates this waterproof barrier, and the grass roots above it can't get any moisture, so they just die of thirst. It creates this necrotic zone.
It's amazing to think about. We walk over lawns every day and think of them as these flat, boring, uniform surfaces. But underneath, there are these silent, expanding cities that have been living there for centuries, leaving these geometric footprints on the surface.
It connects you to the history of the land. When you find a really large fairy ring, you are looking at an organism that might be older than the house sitting next to it. It's incredible.
Okay, so we have our friendly, ring-forming, nitrogen-loving mushroom. History says it is Agaricus campestris. But here is where we have to drop the bomb, because if I'm standing in a field in Ohio or California or Ontario and I say, look, Agaricus campestris, I am, for all intents and purposes, lying.
This is what we call the Kerrigan Bombshell.
Richard Kerrigan, he's the guy when it comes to Agaricus.
He is the definitive authority. And in 2016, he published this huge, extensive body of research that basically upended the table. His work suggests that the true Agaricus campestris, the European species that Linnaeus described way back in 1753, likely does not exist in North America.
At all.
Or if it does, it's incredibly rare. It's probably just an accidental import that hasn't established itself.
So all these field guides that have been printed for the last 50 years, 100 years, they're wrong.
They aren't wrong in a culinary sense because the mushrooms they're describing are edible, but they are wrong taxonomically. It's a classic case of what biologists call lumping. For centuries, we operated on visual confirmation. If it looked like a meadow mushroom and it acted like a meadow mushroom, we just lumped it into the campestris species bucket.
Right.
But Kerrigan used genetic sequencing to show that the North American versions have genetically drifted. They've evolved. They're distinct species.
This is the concept of cryptic speciation, right? Where things look identical to our naked eye, but genetically they've stopped returning each other's calls a long, long time ago.
That's a perfect way to put it. They have evolved separately. So if you are in the eastern United States or the Midwest, you aren't seeing campestris. You are most likely looking at a species called Agaricus porphyrocephalus.
Porphyrocephalus? That sounds like a medical condition.
It's actually a great descriptor. Porphyro comes from the Greek for purple. This species often has these very fine fibers or scales on the cap that can take on a purplish or reddish-brown tinge, especially in the center.
But I thought meadow mushrooms were supposed to be pure white?
They are mostly white, but Porphyrocephalus has that subtle lilac or wine-colored tint to the fibrils on the cap. It's a tell. Then you also have Agaricus argenteus, which is also sometimes known as Agaricus branlii. These are the ones that look almost identical to the European classic.
And what if I'm out on the West Coast in California or Oregon?
Then you're likely seeing a specific variety, Agaricus argenteus variety, Anite.
Okay, so does this actually matter to the person holding the frying pan? If they all taste good and they're all safe, does the name matter?
Functionally.
Yeah.
For the kitchen, not really. They are all meadow mushrooms in that broad culinary sense. They are edible, they taste similar, they grow the same spots. But from a scientific and conservation perspective, it changes everything. It means our biodiversity is much richer and more complex than we thought. We aren't just a copy-paste of Europe. We have our own unique fungal flora.
And it's not just North America. I was completely fascinated by this study from 2024. I think it's source 8 in our stack out of Northeast China. They are doing this deep genetic dive and finding things that completely break the mold.
This is a perfect example of why this field is so exciting right now. We think we know everything about such a common mushroom, and then a team in China analyzes some collections from a forest, and boom, 6 new species described in one single paper.
Let's talk about Agaricus aurantipileatus, because the name alone suggests something really wild.
Right. Auranti means orange, and pileatus refers to the pileus, or the cap. So the orange-capped Agaricus.
An orange meadow mushroom.
Specifically, it has these orange or brownish yellow scales on the cap. It's not, a bright pumpkin orange, but it's definitely not the pure white snowball we're used to seeing. And then there is Agaricus voluminosus.
Volutinous. That means velvety, right? Like the texture?
Exactly. The texture of the cap is like felt or velvet. It's a tactile difference. These are morphological differences that we just ignored or missed for decades because we were so dead set on calling everything campestris.
The study mentions they used multi-gene phylogenetic analyses. This is the heavy lifting. They aren't just looking at the mushroom, they're looking at its genetic barcode.
They're sequencing the ITS region of the DNA, which is the standard barcode for fungi, but they're also looking at really subtle microscopic features that you and I would never, ever see in the field.
Yeah.
For example, they're looking at the Cheilocystidia.
Cheilocystidia, that is a mouthful. What on earth are those?
Okay, so think of the gill edge like a crowded nightclub. Most of the cells on the surface of the gill are called basidia. They are the fertile ones, the ones partying and dropping spores. But on the very, very edge of the gill, you have the Cheilocystidia. They are sterile cells. They're like the bouncers standing at the velvet rope, giving the gill edge its structure.
So they don't reproduce, they're just structural.
Exactly.
Ah.
And in these newly discovered species, the shape of the bouncers is different. In one species, they might be club-shaped. In another, they might be balloon-shaped or bowling pin-shaped. You literally have to put a sliver of the gill under a high-powered microscope and check the ID of the security staff to know which species you actually have.
That is just wild. It shows that what looks like a simple white mushroom is actually this incredibly complex biological machine with specific microscopic architecture that defines it.
That brings us to the so what of taxonomy. Why should a listener care about Cheilocystidia? Because it reminds us that nature is granular. The closer you look, the more incredible variation you find. But for the forager, the specific Latin name matters less than the group name. You need to know you are in the safe group of Agaricus.
Because, as we teased at the beginning of this deep dive, there is a very prominent unsafe group.
There is. And it often lives right next door, sometimes in the very same fairy ring.
This is the twist, the evil twin. Because growing in the exact same lawns, the exact same pastures, looking almost exactly the same to an untrained eye, is Agaricus xanthodermus.
The yellow stainer.
This is the villain of our story. And I feel like the name Yellow Stainer sounds a bit like a comic book villain, but the effects are very, very real.
Agaricus xanthodermus is the most common poisonous look-alike for the meadow mushroom. It is widespread, North America, Europe, Australia. It loves the suburbs. It loves manicured gardens and parks. It is very likely growing in a listener's backyard right now.
And when you say poisonous, are we talking deadly? Will this kill you?
It's not deadly in the sense that it will destroy your liver and kill you in a week like a destroying angel will. It's classified as a gastrointestinal irritant. But that sounds far too clinical and mild. It causes severe violent cramps, nausea, projectile vomiting, sweating, and diarrhea. It is a miserable dehydration-inducing experience that can last for 24 to 48 hours.
And the really tricky thing is, I've read that some people can eat it and they're totally fine.
Yes, there is this weird individual variability that makes it even more insidious. Some people have what foragers call cast iron stomachs, and they can apparently digest it without any issue. But for the vast majority of the population, it is toxic. And you really, really don't want to play Russian roulette with your dinner to find out which category you fall into.
So we need fail-safe tests. We need to arm our listeners with the tools to spot the villain every single time. And the sources point to 3 main ones.
We do. And you need to use them in combination.
Yeah.
Never rely on just one. Rule number 1, the cut test. This is the most reliable visual cue.
Walk us through it. What do I do?
You pick the mushroom, you take a knife, or even just your fingernail, and you cut or scratch the very base of the stem. I mean, right where it comes out of the ground, that little bulb at the bottom.
And what are we looking for? What's the reaction?
If it is a tasty, edible meadow mushroom, the flesh inside should stay white. Maybe after a few minutes, it bruises a slow, dingy, reddish brown. But if it is the yellow stainer,
It puts on a show.
it puts on a spectacular show. It stains a bright, shocking, chrome yellow, think highlighter yellow, and it happens immediately. That pigment is called xanthodermin, hence the name xanthodermus, yellow skin.
Okay, so bright yellow equals bad. White or slow reddish equals good. But there's a catch, isn't there? There's always a catch.
There is always a catch in mycology. There are other toxic species like Agaricus californicus, which is common in, well, California, that also stain yellow, but they do it very faintly or very slowly. If you're impatient and you just glance at it for a second and throw it in the basket, you might miss that subtle warning sign.
Which is why we need a backup test. Test number 2, the smell test. And I love this one because it requires you to really get intimate with the fungus.
You have to. You have to get your nose right in there. Crush the base of the stem a little bit to release the volatile compounds. A true meadow mushroom smells. Well, it smells like a mushroom.
Earthy, Mild, pleasant, like a grocery store button mushroom, but a bit stronger.
Exactly. It's an appetizing smell. But the yellow stainer? It smells of phenol.
And for those of us who didn't spend a lot of time in a chemistry lab, what does phenol smell like?
Think of a hospital corridor. Think of India ink or iodine or Band-Aids. Some people describe it as embalming fluid. It has this sharp, chemical, sweet but acrid scent that kind of hits the back of your throat. It is decidedly not appetizing.
And the sources warn that this smell is a trap, because when the mushroom is cold or if it's a bit dry, the smell might be very faint, but then you cook it.
Oh, the cooking disaster. This is like a classic foraging horror story that you hear all the time. You bring home a basket full of yellow stainers thinking you've scored.
Yeah.
You slice them up, you throw them in a hot buttered pan. The heat
There's also a quick mention of shape in the guides. The blocky cap. Is that reliable?
It's a subtle field mark, and I hesitate to rely on it too much, but yes. Young yellow stainers often have a cap that is more trapezoidal or blocky, kind of flat on top with steep, straight sides, whereas the meadow mushroom tends to be more of a perfect round hemisphere. But shapes change. Wind, rain, and age can distort them. Chemical reactions, like the yellow stain, are facts. Shapes are opinions.
I like that. Rely on the chemistry, not the geometry. Now, speaking of lookalikes, it's not just the yellow stainer we have to worry about. There are other white mushrooms out there. Some of them are actually good, though, right?
Oh, yeah. The Agaricus genus has some absolute all-stars. If you find a massive white mushroom, bigger than a hamburger bun, standing tall in the grass, and it smells strongly of licorice.
Licorice. You mean like anise?
A strong, sweet aniseed smell. That's very likely the horse mushroom, Agaricus arvensis. It's a fantastic edible. It's larger, denser, and firmer than the meadow mushroom. Now, it can bruise a little bit yellow, which is confusing for beginners, but the yellow is slow and kind of a creamy, pale yellow, not that shocking, bright chrome yellow. And the smell anise versus chemicals is the absolute giveaway.
And what if I'm in the woods? We established that meadow mushrooms are campestris of the fields, but if I'm deep in a forest and I see a white mushroom that looks kind of similar?
You need to be very, very careful. It could be the wood mushroom, Agaricus silvicola, which is edible and also smells like anise, but the woods are also the home of the destroying angel.
Amanita virosa, or Amanita bisporigera in North America.
These are the ones that kill you. There are no stomach cramps. It's a little bit of nausea. Then you feel better, and then a week later, your liver fails. They are pure white. They are beautiful. They stand tall and elegant.
So how do I distinguish that deadly Amanita from an edible Agaricus if I'm in the woods?
It comes back to the gills and the feet. The Amanita has pure white gills. They stay white their entire life. They never turn pink or brown. And at the very base of the stem, often buried under the leaves or dirt, the Amanita grows out of a cup-like sac. It's called a vulva.
And Agaricus has no cup.
No vulva for Agaricus. It has a smooth, sometimes slightly bulbous base. So if you are foraging, you never ever just cut the mushroom off at ground level. You have to carefully dig up the whole base, you check for that deadly cup, you check the gill color, and you do the ultimate test, the spore print.
Let's talk about the spore print. This is emphasized in every single guide as the ultimate ID tool. It feels like CSI, Mycology, but it's something anyone can do at home with just a piece of paper and a bowl, right?
It's incredibly simple and elegant science. You take the cap off the stem, you place it, gills down, on a piece of paper. I usually recommend using a piece of paper that is half black and half white so you can see the contrast regardless of what color the spores are. You cover it with a bowl or a glass to stop air currents from blowing the spores away, and you just wait.
How long do you have to wait?
A few hours is usually enough, but overnight is standard. When you carefully lift the cap in the morning, the mushroom will have dropped millions and millions of spores in the precise pattern of the gills. It looks like a photographic negative. It's actually quite beautiful art.
And for a meadow mushroom or any Agaricus, what color are we looking for on that paper?
Chocolate brown. A rich, dark cocoa brown.
And if it's white?
If the print is white, you might have a deadly Amanita. You discard it immediately, wash your hands, and you are very, very glad you did the test.
What if it's like a weird color? Green, maybe?
Green is a very specific warning sign. That indicates chlorophyllum molybdites, the green-spored parasol. It's also known, quite appropriately, as the vomiter. It is the #1 cause of mushroom poisoning in North America because it loves lawns and can look somewhat like an Agaricus from above. But the spore print is this sickly, obvious olive green.
So brown equals Agaricus. White or green equals danger.
Correct. But remember, and this is absolutely crucial, the toxic yellow stainer is also an Agaricus. So it also has a brown spore print.
Ah, right. So the spore print gets you to the right neighborhood, but it doesn't verify the specific house address.
That's the perfect analogy. The spore print rules out the truly deadly Amanitas and the nasty green-spored parasols. But you still need the stain test and the smell test to rule out its toxic cousin, the yellow stainer. It's a multi-step security clearance. You cannot skip a single step.
It really is an exercise in discipline, isn't it? You can't just be casual about it. Nature demands your full attention.
Foraging is not for the absent-minded. It requires you to be fully present with all your senses.
Okay, let's say we've passed all the tests. We have a chocolate brown spore print, smells earthy and mushroomy and not like iodine. There is no bright yellow stain when we cut it. And the gills have turned from that beautiful pink to a nice brown. We have a basket of verified, safe meadow mushrooms. What do we do with them?
You feast. They are considered choice edibles. They're fantastic.
The sources say the flavor is similar to a portobello, but more intense.
Think about it. A store-bought mushroom is grown on sterilized compost in a climate-controlled dark room. A meadow mushroom has grown as actual soil. It's fought with bacteria. It's dealt with the sun and the rain. It has a concentration of wild flavor. It's muskier, meatier, more complex.
But there's a warning about freezing them that I saw in a couple of places.
Yes, do not freeze them raw. If you throw raw Agaricus in the freezer, the ice crystals will shred the cell walls, and when you thaw them, they turn into a slimy, gray, unappetizing mush. You have to cook them first.
How would you do that?
My recommendation is to make a duxelles. You finally chop them, you saute them with shallots and butter and thyme until all the water's cooked out, and you have this thick, concentrated paste. Then you can freeze that paste in ice cube trays, or you can pickle them.
And drying works too.
Drying works very well. They rehydrate beautifully for soups and stews. Or you can grind the dried mushrooms into a powder and use that powder as an umami bomb in gravies and stocks. It's a secret weapon in the kitchen.
And nutritionally, they aren't just empty calories. They're actually pretty good for you.
Not at all. They are surprisingly high in antioxidants. They are packed with minerals like potassium, phosphorus, zinc, iron, and especially selenium. And there is this really interesting note in the medical literature source 10 specifically about potential insulin-like activity.
I saw that. It mentioned that water extracts of Agaricus campestris were shown to enhance insulin secretion in vitro, so in a petri dish.
It's fascinating preliminary research. Now, we have to be extremely clear here. This does not mean eat mushrooms and throw away your diabetes medication. Please do not do that. But it does suggest that these fungi have complex bioactive compounds that interact with our metabolism in ways that we are just beginning to understand.
I want to hit one final safety note on the culinary side, though. We said they grow on lawns.
Yes, the suburban lawn, the modern American battlefield.
Which is often the site of pretty intense chemical warfare.
Herbicides, pesticides, fungicides, fertilizers, not to mention dog pee and road runoff. Mushrooms are not like apples where you can just wash the skin. They are sponges. The mycelium absorbs everything from the environment to build the fruit body. If that lawn was sprayed with weed bee gone yesterday, and you eat the mushrooms today, you are also eating the weed bee gone.
So the location where you pick is just as important as the identification.
Absolutely. It's rule 0 of foraging. You want organic fields, pastures you know haven't been treated with chemicals. Don't pick from the side of a busy highway where car exhaust and heavy metals from brake dust settle into the soil. Use common sense. If the grass looks too perfect and green, it's probably poisoned.
It's funny, we started this episode thinking, oh, the meadow mushroom, the simple one, the one the kids draw. And we've ended up in a world of multi-gene phylogenetic analyses, chemical smell tests, microscopic bouncers on the gills, and toxic lookalikes hiding in plain sight.
That is the absolute beauty of mycology. It constantly challenges the idea that nature is simple or that we have it all figured out. The meadow mushroom is a paradox. It's ubiquitous, it's everywhere, yet scientifically it's elusive. We are still naming brand new species of it in 2024.
That part just blows my mind. We are finding new things in what we all thought was a completely solved problem.
It connects back to that bigger picture for me. Nature doesn't care about our field guides. We write the books, we draw the lines between species, but the fungi are out there doing their own thing, swapping genes, adapting to new environments, and evolving right under our noses.
So what does this all mean for the listener? The next time they are walking across a park or their own lawn and they see that little white mushroom popping up in the grass.
I want them to stop. Don't just kick it over. Don't just ignore it. Crouch down. Look underneath. Are the gills pink? Is there a ring on the stem? If you scratch the base of the stem, does it glow yellow?
Engage all the senses. Sight, smell, touch.
Exactly. Even if you never ever eat it. And frankly, until you are an absolute expert, maybe leave the eating to the experts, just engaging with the complexity of that one single organism changes how you see the world. You realize that a lawn isn't just grass. It's a battlefield. It's a city. It's a biological mystery waiting to be decoded.
And a final provocative thought to leave you with. If the standard field mushroom that's in all our old books might not even exist in North America, and we are just discovering new species in our own backyards now,
Yeah.
how many other common things in your environment are actually total strangers to science?
We are just scratching the surface. It's a whole world down there.
Thanks for diving in with us. Keep your eyes on the ground.
And your nose ready for the smell of iodine?
See you next time.